Hello, everyone. Welcome to today's Ask the Expert panel. This expert panel discussion is presented by LabRoots and sponsored by Gibco, part of Thermo Fisher Scientific. Today's panel will include the following experts: Richard Altman, scientist, protein technologies at Amgen Incorporated; John Zmuda, Director of Cell Biology, Life Sciences Group at Thermo Fisher Scientific; Konrad Majkowski, scientist, protein and cell sciences at EMD Serono Research and Development Institute. And I'm Henry Chu. I'm Associate Director in Cell Biology, Life Sciences Group at Thermo Fisher Scientific. And I'm going to be your moderator today. So before we begin, I'd just like to remind everybody that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during this panel discussion. Just click on the green Q&A button located on the lower left of the presentation window and type your question into the box that appears on the screen. We'll try to answer as many questions as we have time for at the end of, during the panel, this live panel discussion. Please also notice that you'll be viewing the presentation in the slide window. Actually, all you'll see is I'll be switching back and forth from the different panelists as they speak, so that you can just see who's talking there. And to enlarge this window, if you'd like, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. So just to begin this Ask the Experts panel session, what I'd like to do is just take a few minutes each and let each of our panelists, Rich, John, and Konrad, introduce themselves and tell a little bit about what they do, particularly as related to protein expression work. So Rich, let's start off with you and then go to John and then to Konrad, if that's okay.
Sure. So hi, everyone. My name is Rich Altman, and I'm here at Amgen in San Francisco in the Protein Technologies Group. I've been doing protein expression for probably about 20 years at this point. We have a group here that is using both transient and stable expression and a variety of cell lines in the transient realm and a variety of scales to support multi-site drug discovery efforts. We're running scales anywhere from 24, a couple of mils in a 24 deep wall plate to weight bioreactors. And our focus on, a lot of our focus is on, making our processes more efficient. How are we choosing expression systems, and how are we assembling our expression toolbox to meet those needs?
Great. Thank you very much, Rich. John, could you introduction?
Yes, thanks, Henry. For those of you that happened to be online earlier today to see my talk, my name is Jonathan Zmuda. I'm a Director of Cell Biology at our Frederick, Maryland site for Thermo Fisher, about 30 minutes due north of Washington, D.C. One of the prominent roles that my team plays here in terms of developing new products and technologies is with the transient expression platforms, both insect as well as mammalian 293 and CHO. And one of the things that we are continually looking for is to understand the as yet unmet needs of the protein expression community. I think one of the common themes that we all see is that no one size fits all. And to use Rich's terms, what we look to do is develop a toolbox of technologies so that at some point in the hopefully not so distant future, whatever protein it is that you need to make and whatever quantities that needs to be made in, there will be a tool and a high expressing system to allow for that work to take place.
Thank you very much, John. Konrad, I turn it over to you.
Hi, my name is Konrad Majkowski. I'm a scientist at EMD Serono. I started almost exactly a year ago here. And prior to EMD Serono, I worked for 25 years at Biogen and a few smaller companies in between. I spent most of my time expressing protein from various cell lines, insect cells, mammalian cells, CHO hybridoma. Right now at Serrano, I'm in a group that is responsible for delivering protein to our projects at high throughput and small scale, medium scale, and up to 10 or 20 liter scale. That's our goal. So anything between 15 mils and I'd say 10, 20 liters. That's our role over here, and we're using predominantly XP293 and XP-CHO system, as well as some CHO as stable clones.
Great, wonderful. Thank you very much, Conrad. So at this point, then, we are going to open it up, and this is, again, Ask the Experts panel, so we appreciate as many questions as we can. We already have a few, but for anybody on the line, you can go ahead and start to enter in whatever questions that you might have. So let me just start with a couple here that we've already received in. The first one here I'll ask is, for you three who are, have done a lot of work, and I think, in both 293 and CHO cells, what type of differences you typically observe in proteins if you express them in 293 cells versus CHO? So let's start with Rich, if you wouldn't mind just, giving your thoughts on that, and then we'll go from there.
Yeah, I think probably John and Conrad will agree with this, but the major differences that we see is about the glycan profiling, and so which I think, as I mentioned during my talk, that I think that system has opened up a lot of efficiency for our processes in being able to do that. And I do think, as I mentioned before, that, again, depending on the protocol that you're running, you're seeing a much more production-like glycan profile than you do in the 293s. But I'll always throw in the caveat is that I love the XP293 system, and that it is really a workhorse and provides a lot of proteins in a lot of different formats, and successfully meets the needs for a lot of requests that we have.
Okay. Great, thank you. Observations from Conrad and John?
What I could add is, I think Matthew McKenna added also that, and I totally agree with that, is that the 293s have much shorter cycles. So if quick turnaround is the thing that you need, you can exploit that 293 quick expression to get the benefits of the higher titer from XP-CHO. You have to wait 7, 10, 11, sometimes 14 days, but that depends what's the goal. If the goal is to get as much protein as possible, then XP-CHO in many cases is the preferred system. If the speed is what you need, then 293 maybe more of a benefit to you. One more thing that we, for example, take advantage of is 293 media. It seems to be a little bit more IMAC columns friendly. We're finding less chelating, or at least our scientists in downstream processing find a way to work around it, while XP-CHO media seems to be causing a problem with using nickel columns.
Okay, great. Actually, you were sort of answering a little bit of another question that we have in here, so maybe I'll just go ahead and throw this out here now as well. And again, maybe John, you can start off, but certainly it's great to see. Oh, and I've been forgetting to switch to pictures. Sorry about that. But to just get the interaction between you guys to see what you say. But here's a question another question that came in. So said, I'm interested in expressing GST or his-tag protein fragments for purification and subsequent antibody production in mice. So what system would you recommend to produce up to 10 milligrams of protein with native glycosylation patterns? So, John, we'll let you start off, if you don't mind.
Yeah, great. Thanks, Henry. Just to go back to that last question very quickly, I agree very much with both Rich and Conrad on many of those points. The one interesting thing I think with regard to the 293 and the CHO cells in general, in their terms of their biology, is that the 293 cells really have a quick burst of protein expression, and then they fizzle out. And very different than the CHO cells, and you can really see why the CHO cells are so valuable as a bioproduction cell line in the manner in which that they ramp up their protein expression and then are able to just continue to pump out protein as long as you keep them healthy in the culture. So very different than the 293 cells. And similarly, along those lines, what that allows for is some flexibility in terms of how you perform the protocols. So, for example, in terms of, to get protein quickly, in many instances, the rate of protein XP-CHO can be similar to XP293, but also there are ways that modifications to XP-CHO in terms of transfecting at even higher cell densities, so very high densities at 10 to 12 million cells per mil, that will allow you to make protein even faster and can really rival or even outpace the 293 system if you're looking for about a 4-day turnaround time from the system. And then lastly, just in terms of the differences in the protein, one of the things that I think is still just coming about right now of proteins that express well in CHO cells transiently, and being able to apply that to whether or not those same constructs or signal sequences, et cetera, will be successful in the stable CHO production. So while that's still sort of a new and developing field, there's some information that you may certainly have some of that information bleeding through to the stable cell line expression as well. In terms of the GST or his-tag protein expression, I think right now, as Rich mentioned, XP293 has been around for many years and has been a workhorse for a lot of these proteins, and probably has a larger data set for non-antibody proteins currently than XP-CHO. But I think in general for both of the systems and either one, utilizing the correct resins for doing the nickel purifications, for example, is critical. All of the high-density medias, and including the feeds, have componentry that can impact nickel purification. And so, for example, some of the strip-resistant resins, such as the Excel resin, et cetera, we've heard have been, and we've tested, been very good at recovering his-tag proteins from the various supernatants.
Great. Thank you. Any Rich or Conrad, any experience on your end in terms of tags and purification and what works well for you in the work that you do?
From my point of view, it's like I said, our downstream processing group, the purification group, feels more comfortable with dealing with media from XP293, and they dictating to what expression system. If we're expressing Fabs with his tag, they prefer if we're expressing in 293 system. I guess at that point the glycosylation is not an issue for them, and they just want to have it expressed and easier to purify it. I will address and I will tell them about what John said and challenge them if they fully exploited the XP-CHO media, if that can be modified to purify it on the same resins. And I totally agree with John that this new ultra high density system is very good substitute for 293s.
Thanks.
Yeah, and I think that speed issue, and we keep coming back to that, but I think that just continues to drive a major way that we're thinking. Getting those XP-CHOs, and we too have successfully done that a number of times at that 4-day. And John's exactly right. What we see is at 4 days, a lot of times titer is equivalent or superior to what we see in the XP293s. And also, I think one of the things, and it's sort of we're still sort of working on it. And I don't know, John, maybe you have some more comments about this, but in some of these format issues where we might see some issues with product quality, maybe that shorter duration helps us not only in just the process time, but maybe in the product quality time.
Yeah, absolutely, and I covered that a little bit in my talk this morning. That for depending on the downstream use of your protein. So, for example, if you're making an antibody as a reagent, and what you need out of that is you need as much as you can get, and it has good binding qualities. Certainly, you can push in ex vivo all the way out to 14 days. 12, 14 days, make as much protein as you can. Should be intact and stable. If you're trying to match glycan patterns, for example, to a production cell line, that's something that, again, you want to take a look at. Rich was one of the authors in some very early work done with XB293 and XB-CHO, showing that compared to a stable cell line, that expressing an XB-CHO at 37C, compared to that particular stable cell line, generated glycan patterns that were most similar to the stable cell line. So understanding, again, what the intended purpose of the material is very important, and then tailoring your harvest time to meet that. And then, outside of antibodies, non-antibody proteins are going to have, different profiles, both in terms of their impact on cell viability, as well as their post-translational modifications, stability, clipping, etc. All of those things, have to be taken into account, sort of on a per protein basis, and determine how far out you're able to go in terms of the trade-off between titer and activity and structure function. And then lastly, when you talk about proteins, either toxic proteins or membrane proteins or unstable proteins, you may be looking at harvesting at 24, 48, 96 hours. And again, that's all based on the nature of the protein. So if you think about sort of these three different classes of proteins, it gives you a fairly good idea of where that starting point is in terms of where you should be looking to harvest protein. Again, taking into significant account what the downstream applicability of that protein will be.
Okay, great.
Yeah, I think that's a great point, John, just in the sense that in terms of you brought up about membrane proteins, about how that's probably for most of us becoming a higher and higher percentage of things that we're expressing, and how we have to sort of alter our harvest time in some of our conditions to sort of coax the most out of that we can.
And Rich, to follow up on that, we are in the process of doing some fairly significant collaborations around membrane protein expression with a few different outside experts. And we have recently expressed some proteins in both XB293 and XB-CHO, GPCRs in this case, using pcDNA vectors, so 3', 3'4, etc. And one of the strategies that has seemed to so far been fairly successful has been even using these high expressing CMV-driven promoters. We've been able to significantly reduce, in the case of XB-CHO, the DNA going into the system, so down to perhaps 0.2 or 0.4 micrograms of plasmid DNA per mil of culture media to be transfected. And we've definitely seen that those lower amounts of plasmid DNA are providing higher expression of the membrane proteins. And in these preliminary studies, we're seeing at XB-CHO that both the expression levels as well as the functional activity of these GPCRs are equivalent, if not better, to what we're seeing in 293 and bacterially expressed GPCRs. Again, this being a small data set right now with a significant amount of additional data to come later this year.
John, on that subject, when you reduce the amount of DNA for the membrane expressed protein, right, do you also reduce the amount of X-bifectamine proportionally?
Yeah, so that's a great question, and this is another significant difference between the 293 and the CHO system. The 293 transfection reagent works best right around that 1 microgram of plasmid DNA, and with XB-CHO, you can use significantly less plasmid DNA. We showed some data earlier today going down to 0.2, 0.4 micrograms. In certain instances, you could certainly even go lower than that. What I would suggest for XBCHO is that if you're operating below about 0.6 micrograms of plasmid DNA, so if you go down to 0.4, you reduce the amount of your transfection reagent proportionally as you continue to scale down. That will actually allow you to utilize the smallest amount of DNA to generate still very good titers of proteins. And so, yes, it does become important to, at some point, and again around 0.4 micrograms, to proportionally lower the transfection reagent as well, so that you have a proper stoichiometry of the complexation reaction. But we continue to do the entire complexation reaction in the normal volume that you would typically use for the protocol, just much less DNA and transfection reagent.
And John, have you tried the? So you're reducing the DNA, but do you add, sort of, we go back to the gene dosing, do you add, or have you done empty vector or something like that to keep the amount of DNA constant?
So at this point, we have not done that. So I think that is a very well-known strategy, and that allows you at that point to not have to manipulate the amount of transfection reagent. It keeps the whole complexation reaction as is. And there may be other benefits there as well. If you're looking to slow down the expression of the protein, it's possible that those empty vectors could soak up some of the transcription factors, slow things down a little bit in the cells. So I think that's an excellent strategy. We recently had a case study for a difficult to express protein where the group expressing it was using XBCHO. They significantly reduced the DNA level, tested it at the two different temperatures, and in fact were able to find a combination that worked very well with the temperature shift, but actually not as well when it was at 37C. So for some of these difficult to express proteins, we've certainly seen instances where some will express better at 37C, which typically, if you look at the XBCHO protocol, that would be thought to be the lower expressing temperature, but certainly some of these proteins express better at 37 and some at 32. So when you run into these tricky proteins, it's always good to take a look at both of the protocols and see which one is generating the best data.
Great. Hey, thanks a lot, guys. Great response to the questions as well as additional work. Additional discussion there. You can see that when you get a group of scientists together to talk shop, that a lot of interesting points come out of that. Let's go. I'm going to go ahead to another question, and just for anybody who's joined a little later into our Ask the Expert panel, just a reminder that we're completely open at any time. If you click on the Q&A button, and down at the bottom you can write in a question, and then hopefully we'll be able to get to that. So here's another question that came in. I'd like to ask this. This person is asking, should I re-optimize expression vectors that I use for transient expression in 293 cells to make them more specific for expression in CHO cells? And how much more yield potentially could I get if I were to do that? So Conrad, I don't know if I know you work with both systems. If you'd like to at least start off and whatever your observations are about that.
I actually, in our case, it's all about speed. So we don't optimize if something does not express well. If we switch from one system to another, we just make more. I think re-optimizing vectors would take too much time. So we do not have any optimized vectors for 293s or XBCHO. We use the same DNA from one to another. If it doesn't express well in 293, we switch to XBCHO using the same plasmid DNA.
Okay, thanks. Any observations on your end, Rich or John?
No, so I guess I would second what Conrad's saying, that I think one of the advantages in these systems is that the systems are pretty agnostic to the vectors, and so I think we've taken full advantage of that over the years. And I think from trying to get to a point where we minimize the number of vectors that we have, both for transient and stable cell line generation, it sort of plays into that we can sort of go in between systems without recloning or doing any of that or creating more constructs up front. So sorry, the long-winded answer to that is no, we don't. We just sort of switch between systems using the same vector.
And then I'll just add one small detail there, a little bit offshoot, but typically we found in 293 cells for many years now, the thought has been to use a heavy chain to light chain ratio of about 1:2 for best expression. Whereas we and many other groups seem to have found that in the CHO cells, a 1:1 heavy chain to light chain ratio in the vectors seems to be most optimal, as the CHO cells seem to do a very good job expressing the light chain. And lastly, when it comes to vector optimization, there's an awful lot of anecdotal information out about different vectors, different systems, different proteins even. And, that I think it's very tricky to come up with a definitive conclusion around, which vector or promoter is going to be best for a given protein in a given system. And to Conrad and Rich's points, you can chase vector design around for a long time, but if you need to get protein made, that's unlikely to be the successful strategy to do so in a reasonable amount of time.
Yeah, I think the whole idea of transient system is quick delivery and don't mess around with it, so everything is just compartmentalized, and you go with what you can, double up the volume if the titer is not big enough, and move on.
Yeah, and John, I totally second what you say about the ratios. That's much easier to do if you're utilizing two vectors for that in terms of the heavy chain, light chain. We find the same thing about 2:1 light chain to heavy chain. Or I guess in the past we've also done iris elements and put the heavy chain behind the iris to sort of modify that if we have a single vector. But yeah, I'm totally in agreement with you about that.
All right. Thank you very much. We have a question here. I know, most of the work that people do in transients are done in shake flask. So this person had a question just about scaling, and particularly scaling up. So the question they have is, when do you think it makes most sense to, when you're scaling up your cultures, I guess, in this context, when do you think it makes most sense to switch from large shake flasks to go into bioreactors when you are trying to make protein in larger volumes? John, do you want to start off with this one?
Well, actually, I'm going to defer that one. Conrad and I were just talking about some of this work earlier. Conrad, do you want to start off on that, since I think you deal with that issue on a weekly basis?
Yeah. So I'm kind of like in two worlds in where we are. We either do things on a very small scale, like literally 15, 20 mils, or the next step is a liter and up. And then recently, like November, December, we started to work on a waveback in collaboration with John and his lab, and trying to figure out waveback and stir tank bioreactor. We didn't have much success with optimizing bioreactor, but we feel like we kind of have waveback figured out, and that has been a big breakthrough for us. Now we can, between the high titers and the larger volumes we can do, this becomes a very good system to express actually gram quantities of protein for larger studies. So it depends what the needs are, but when you combine system that is capable of expressing over a gram per liter, and if you can optimize or dial in your large scale expression, that's a very powerful tool. So it's all needs driven, but the new protocols that are coming up for waveback and for large shake flask are pretty much mimicking what the expression is in the spin tubes and small shake flask. So I'm no longer afraid of the large volume.
So for you, when do you, what's the cutoff point or the volume at which you typically are saying, okay, I'm going to move over to the bioreactors, the waves, rather than in the shake?
So usually in our case, we do screen, we do high throughput, multiple constructs, and then we get a feel for how much they express in a high throughput scale. And if the titer dictates that, for example, it expresses 300 or 500 mg per liter, and I need 5 grams, then I know I'm gonna need 5 liters of it. So at that point, I can either do 5 large shake flask or wave back. Pretty much at this point, we have overlapping scale that I can go from spin tubes to medium-sized shake flask to large shake flask, and then overlap to wave back. So it's all it's all driven by the request right now.
Okay, thank you. Rich, any thoughts on your end, or how do you typically make decisions in this way if you do scale up work?
Yeah, no, I think it's a really good question. That I think you get in that range because of the advances of some of these consumables. Between the Corning 5-liter flask and the Thompson 5-liter flask, has sort of expanded what you can conveniently do. Again, depending on your incubator space, I think you get up into that. You can easily handle the 5-liter scale. It's sort of a breaking point. You can go onto a wave where you can keep it in the flasks, and that's pretty easy to handle. I think probably most of the time now we reach 10 liters, and then we go into the waves, and we're using waves for both stable scale ups and transients and multiple systems. I think I agree with Conrad in the sense of if you have one of these where you need gram or multi-gram quantities, the wave is a great choice, especially if you have good titer. But the other thing we're seeing is also one of these where you have a low expressor and you need half a gram, and so the wave becomes really an important tool there too. But also tweaking the conditions in the wave. We just had a recent case where we had a pretty low expressor, a non-antibody. It's 8 or 10 mg per liter, and we needed 500 mg. And so, with some optimization in the wave, we're up to in the range of 50 to 70 mg. So that was helpful. And I think those lower end ones, those lower expressors but higher quantities, relatively speaking, are also make up a number of our requests these days. So. I think one of the things that, and maybe Conrad can weigh in, and John as well, is that, how do you set that up? It's nice to be able just to seed the bag and expand in the bag and transfect in the bag, and now you're down to one vessel doing that versus maybe scaling up in flasks and then, having to transfer all of that material on a volume basis into the wave. But I think to get back to the question, though, that once we get above 5 liters, we pretty much defer to the wave. Although I was a little disappointed to hear Conrad in the bioreactor, because we have a couple bioreactors with 2.5 and 5 liter working volumes downstairs that I want to play with. But I think we have a good bridging mechanism between those flasks and the wave at this point.
So you were successful in a steel tank bioreactors?
No, I haven't tried them yet, and so it's one of those things where it's just hasn't breached the to-do list up into a critical area, and so. But I'd like to, and I think that is a space. I don't know what size your bioreactors are. These are relatively small, but I think it represents an interesting space in terms of volume, what we could do. Probably realistically, I look at those bioreactors as something that probably helps if we have a stable versus a transient, just in terms of the better control that we have over them to create a higher density and more productive. But I still would like to explore them for transient use in that sort of 2.5 to 10 liter space.
So where I see the bioreactor being useful, because there were two reasons. I tried bioreactors that we had on loan over here, and I tried 2 or 3 times, and at the same time I was working with wave, and initially I could only get small shake flask and then medium and large shake flask working, and later on we figure out how to do wave work. I could not have any more shots at the bioreactor, but I failed first 2 times, and I still don't know what, because all the parameters were correct for the transfection. I think I learned much more since then, but I don't see much of an advantage for the stir tank bioreactor versus wave bag, because they are relatively very forgiving with what you can do in a wave. But now I'm more and more of a fan of the ultra-high transfection, ultra-high cell density transfection, and this may be a place where if we can figure out how to set up the bioreactor for regular transfection, that sparging capability of bioreactor may be something that is needed for those ultra-high density transfection. Other than that, I'm very happy with the wave bag, and I don't need to go in a stir tank unless we're talking, over 50-liter scale.
Yeah, I think that's a great point, because I think when you talk about going into bioreactors, the waves, even the new Wave 25s, are so straightforward to use, and although you have more control, but I think you're looking at an additional intellectual investment into really becoming facile in the more stir tank bioreactors. And so is it something not only just from a process, but in terms of really grasping all of the engineering principles in those bioreactors? That's a whole different level of investment, and I think that just depends on what you're using it for. But keeping it simple and keeping the turnaround time easy, it much more favors waves, I think.
Yeah, and I think on our end, obviously, we're looking to support the protocol development of both wave and stir tank bioreactors, and many of the principles that we've identified, using the ready-to-process wave units with the control parameters and readouts. We believe that, implementing some of them to the stir tank bioreactors, of which we have work ongoing as we speak, we hope that some of those changes. So with XBCHO, we found that you have to, in order to scale up to the waves, there's some very slight protocol differences versus the shake flasks, and just in terms of how the cells behave in the wave bags, the different levels of control, oxygenation, etc. And we look to put those principles into play in the stir tank bioreactors as well. And Rich, to your point about growing everything up and transfecting them directly, in fact, in the work that we are doing currently with the wave sort of final optimization experiments, we do either seed at 500,000 or 1.2 million for either 2 or 3 days, and just slightly overgrow the cells, and then add some fresh media at the time of transfection to bring them down to 6 million, such that it's a very easy and straightforward inoculation in the wave bags.
Right. The one thing I'd like to add to it, and I'm in the process of growing 2 of them, and I'm using the 10-liter bag, and it's hard to go low volume in a way that I'm actually not comfortable with going low in a way that I would grow cells to like 8 or 9 millions, and then add fresh media to bring it down to 6. So what we do, we actually grow them at 4 liter, and on day of transfection, we drain some of the cells, add fresh media to dilute them down to 6 million, and we do transfection in a flask, in a hood, and then we just weld the flask on, and we add it. And when I was talking with John's scientist, the question came up: can I make it quick enough to be under 5 minutes? And I actually timed myself, and I can add DNA and all the XP-vitamin and everything in a hood, put a cap on, go to my wave, weld it on, and within 2 minutes I'm dripping. In less than 5 minutes, everything is in.
And are you actively? Oh, sorry.
Go ahead, Rich.
I was just going to say, are you actively pumping that into the bag, and are you concerned about being harsh on the cells at that point, or is it just you're letting it go in by gravity?
No. Cells are in a wave. So cells are grown in a wave bioreactor, kind of like the way John described it. We overgrow them slightly.
I got you.
and drain what's needed, replace with fresh media down to 6 million. And then I do my DNA exifectamine mix in a hood, go to the bioreactor, hook that up, and just drain it by gravity.
Gotcha. Thanks.
Yeah, and using the flask with transfer caps also works well. In some instances, we've had some issues with the tube fusers, and that can lead to significant delays. I think in general, the team here has been utilizing the transfer cap flask to just gravity drain, and in that manner, able to very easily get the full transfer within certainly within 5-minute time period.
Yeah.
Okay, thank you very much, gentlemen. Here's a question here, just, so what features do you think that you'd like to see in a next generation 293 expression system? What do you think are the things that could be further improved at this point? Let's see. Let's start off, John, what are your thoughts on this? I know obviously we're together. You're doing a lot of development work on expression systems. What are your thoughts?
For a next-gen 293 system, I think, clearly the needs are always around, more protein while maintaining quality. And again, the biology of the 293 cells being very different than the CHO cells, makes that a difficult task to continue to improve those titers. But certainly that's something that we would all want to drive towards. And then also, for example, there's a lot of small nuances. So XB293, having been out for many years now, we have generated a lot of great feedback from the industry and academic partners around small things. So for example, the notion of his tag purifications that had come up earlier. So there's probably still an awful lot of areas where tweaks along those lines could be very useful to making it a more, an even further rounded system. Then on top of that, certainly I think there's interest in branching out these systems into structural biology, so potentially some, cell line engineering that imparts characteristics to proteins of interest, et cetera. That I think will build on what is already a very solid expression foundation and move some of these systems into even further utility than where they are today.
Great, thank you. Rich, what do you want John to work on?
Is it just one item I get to choose, or can I have a lot? So I agree with John on all of that, what he said about the 293s, but I think the other thing is, John and I and you too, Henry, have talked about this in terms of, can you do a higher density transfection in the 293s and pushing it like, have some sort of equivalent like you're seeing with the XB-CHOs. Because certainly, as we pointed out, even though it's been around for a while, it certainly is a really valuable tool and quite a workhorse. But I think pushing that and to your point, John, getting to more protein, could we do that and even have a shorter duration? That would be great. I still say the pressure's on to make these run durations as fast as possible.
Great. Any what's on your wish list, Conrad?
Well, I'm in love with this very short coupling time. So if that 15 minutes can be reduced down to 5 minutes, that would be great too. So that would probably require changing the chemical that the fectamine part. But that's something that is slowing me down a lot in the hood when I have to wait 15 minutes for this complex to be formed. So if that can be shortened to the same way that XB-CHO are working, that will be great improvement from our point of view, because especially at the high throughput scale, you do 1 rack, 2 racks, and then you can't really do anymore because you have to wait for the first one and second one to kind of get to the part that you can add the second, you can add cells. So if that can be speed up, that would be great.
Great, wonderful. Thank you very much. We're coming on to about 15 minutes left in this session. So again, another reminder, particularly for anybody who might have joined a little later, that this is an open session. So any questions that you'd like answered, things that are high interest to you, please feel free to type in your questions into the Q&A box and submit those. I've got another question here. So this one is, are there any particular tricks or strategies to maximize expression of chimeric proteins, such as bispecifics, Fc fusion, various other more specialized formats like that? Any volunteers to take this on? I'm not sure who has the most experience with doing any of these types of work.
So I can, just off of some information that we've gathered, over the years, certainly we have seen that the CHO cells oftentimes will do a very good job of expressing some of these more odd and unnatural proteins. So we have specific examples in which bispecific type molecules have not been able to been expressed in prior CHO or 293 systems, and in fact in XB-CHO we're able to see half a gram, 3/4 of a gram per liter for some of these proteins. One of the things that we've heard often is that certainly in the case of, for example, Fc fusion proteins, some of the experts in the field have suggested that in terms of the ability to express them intact, that the secretion signals can be critical for Fc fusion proteins. And in fact, the secretion signals and the manner in which that the proteins are passing through the secretory pathway of the cells can be critical in generating intact and unclipped Fc fusion proteins, such that a construct that would initially potentially be clipped somewhere in the fusion partner with an optimized secretion signal actually can be made to express intact. So that's some very interesting information along those lines. So I think this one is still a topic in development, and I'll certainly turn over to Conrad and Rich, who probably have better access to some of these constructs in day-to-day testing.
Rich, you want me to go?
Yeah. So I'll follow up with John about that. I do think that's interesting. I think some of the things are pretty traditional. I think we come back to when I think of some of these bispecifics or whatever, it's sort of an interesting dialogue between the protein engineers and the expression people, and each of us asking each other, what can you do for us in terms of helping us out when we see product quality issues with some of these? And I do think it's sort of both sides contributing to solutions about this. I do think in the past I've seen when we've had some of different formats, some of these bispecifics or fusions that are prone to aggregation. I've seen the gene dosing being effective in terms of getting a better quality product, and then of course the typical sort of things about temp shift or run duration, of course, are things are certainly things to try as well. I don't know, and also we've looked at basically changing some of the sequence parameters in terms of linkers and linker lengths, and this has been over the course of years and years at different places, and that too can have an effect. I think we're all in agreement about the use of our time, and I think it just depends on the how strong your that particular construct or that particular target is, with how much effort you go to doing all of these sort of things in addition in terms of signal sequences or gene dosing or any of these. So I think that's the other factor to keep in mind. I mean, some things Conrad probably can attest to as well. you need to have expressed no matter what, and others are like, okay, if it's not expressed, this becomes sort of a selection criteria in the whole process. So that's sort of some of our thoughts about that.
I agree with all of that, and another thing I can add, although this is kind of more like in many ways anecdotal, is in my experience, a lot of this difficult to express protein express better at lower temperature. It does something to the cells. They kind of like seems to be taking their time expressing the protein, and in a way the titer is higher because they express better. They assemble the protein better. So we found that often the difficult to assemble protein are expressed better at 32 degrees, which would indicate that the XP-CHO system, when you do the temp drop, is better system. I don't have data showing that if I use XP-CHO at 37 degrees versus XP-CHO at 32 degrees, the XP-CHO 37 is going to be similar to XP-293s, but that's the trend over here, that if we have a difficult to express protein, we're moving it over to XP-CHO and a temp drop.
Yeah, and John, I'd like to follow up, and I might even include the moderator in this because he's been kept out of the fray. But what do you see in terms of sort of cell line engineering with some of these things? I mean, do you see some engineering in terms of some of the ER proteins, in terms of chaperones or folding or those sort of things? Is something in the future that will help this along?
Yeah, I think that the possibilities really are endless in terms of cell line engineering, and I think one of the things that we're working towards again is to have solid foundations of high expressing systems, multiple different host cells, including insect work that's ongoing as well, that provides that initial foundation. So you've got 1, 2, 3, 4, maybe 5, who knows, high expressing systems that give you that first shot at getting the protein that you need. And then, the engineering path, I think there's, certain pathways that can be relatively easily modulated through cell line engineering to provide, for example, protein that is better for crystallography, or, potentially ways to improve the expression of toxic proteins through the modulation of the timing of expression. So there are certain things that, you could imagine would have sort of a wholesale approach or a wholesale benefit to expressing certain types of proteins for certain uses. And then the other issues around, overexpression of various chaperones or anti-apoptotic molecules, et cetera, those become a bit tricky in that, are they molecule specific? Do you need to express a certain set of chaperones for, each one of these difficult to express proteins, and does it then become even feasible to have engineered cells that more or less are, capable of handling every hard to express protein? So, there may be strategies there in terms of cell line engineering, as well as potentially other as yet unknown host systems that may, be less impacted by some of the very atypical molecules that are going into clinical trials today.
Great. Thank you very much. Looks like we probably have time for probably one, maybe two more questions in there. So let me give you this one here. So do you ever see any problems getting expressed proteins to be secreted from 293 or CHO cells, particularly in a situation where the protein is fused to, for example, a GST tag for subsequent purification work? I'm not sure if any of you have some experience with that, either GST tags or having difficulties with expressing or getting secretion of your proteins.
I mean, I can certainly start with an interesting anecdote that came from one of the roundtable discussions recently at the PEGS Boston conference. From Dom Esposito over at Lidos, and this question came up, and Dom's response was very interesting in that anytime that you're trying to take a protein that isn't secreted and doesn't naturally move through the secretory machinery of the cells, you can be asking for trouble, because that protein obviously is going to be acted upon by many different enzymes, et cetera, that it normally is not expected to associate with. So there may be significant post-translational modification changes in trying to drive a non-secreted protein to become a secreted protein. So I think that one is something that you weigh those potential risks and what may be changing with the protein when you're trying to push it out of the cells, which obviously is a more streamlined approach to producing the proteins. But I think one has to be cautious also on what you may be changing by having the protein expressed through the secretory pathway, where it is not typically exposed to in natural expression.
Thank you. Comments, Conrad?
I don't have an experience with expressing GST tagged protein in 293 or CHO cells. I've done a lot of expressing with GST tagged protein, but they're all in insect cells. And the only thing that I always remember that GST helped me to tag and purify it, but very often when the tag was removed subsequently, the protein crashed out of solution because, like John said, that protein does not like to be the way it's constructed to begin with, and GST stabilized it, but then once the tag was removed, the protein crashed out of solution anyway. But that's totally different issue. But in terms of expressing this protein in 293 or CHO cells, I don't have an experience with GST tags. Plenty of his tags, no problem here, but no GST tags on my end.
Thanks. Any last comments from you, Rich, on this?
No, I'm with Conrad about that one. I think it's an interesting question just in general what the efficiency of secretion is, because I think a lot of times we get the result, but it would be interesting more than just sort of sporadically when you run into a problem that you think something is lower titer than what you'd expect, and you go and look and see is it stuck in the cell or whatever, but what the what sort of are acceptable levels, or what should we expect in terms of the secretion efficiency. So, but I haven't done tags in either one of these systems, GST tags in either one of these systems.
All right, thank you very much. I think in the last 4 minutes, this last question I'll ask for you that was submitted in here was, so in your experience, when you're using transient work and doing culture volumes of 1 liter or more, what's been the best way that you've had to clarify that culture supernatant to then move it on for purification work? Rich, maybe you can start off there since I already have your photo up there.
Okay, so this is something that is sort of dear to everybody's heart, because I think it just takes a lot of time. But we're looking at a couple things besides centrifugation now in terms of using these dual filter units from GE in terms of being able to just plug and play, just hook these up to the bag through the sterile connections, start pumping it out and be able to walk away for the hour or two that's required for it to go through the clarification and then sterilization filters in there, and then end up in a sterile bag. And that we've had some reasonable success with. I still think it's a work in progress, but it certainly frees up some time for us to do other things. I think we are also investigating right now the use of magnetic beads that are coated with protein A or His to drop into the bag, say into a weight bag the night before harvest, and then the next day using a magnetic rod basically to pull those beads out and to move them from basically from one container to another, where you are then washing and then eluting off in a concentrated way, is that it'll take about an hour and a half that we now have gone through a clarification, a first step purification, and a concentration step is really exciting to us, and even better in terms of saving time. But That's especially helpful, I think, when you get above, a few liters or above 5 liters for us, that gets us away from the centrifugation step.
Great, thank you very much. Any last very quick comments on this clarification question there, Conrad?
So I agree with Rich. That I saw the presentation at San Diego, and those magnetic beads that was great, and the way they were put in, and the system they built around it, that looked great. Very, very promising. You kind of ignore all the cells and you pull everything at once. The only thing I can add is I've been experimenting with Thomson's this new filter that is the screw-on filter, and that seems to be working well for me as well. It has some limitations. On one hand, it's the size for certain density, but it's a great system that you can just screw on a flask and pump it out of it. I guess the benefits of it, or the trick to it, is that by vacuuming the cells on rather than pumping on, creating pressure vacuum, it increases the throughput. Other than that, we on a smaller scale, we just centrifuge hard and filter it using various devices.
All right, great. Thank you very much. Yes,
Henry,
John, you got 30 seconds. Okay.
Yeah, just chime in very quickly. We are also looking at alternatives that I know other groups are as well, in terms of the utilization of diatomaceous earth on top of the filters, which can allow for pouring cell-containing supernatant directly over filters. Does a nice job of pre-processing the samples, as well as other bioproduction types of filtration mechanisms, including flocculation of the culture supernatant to remove a lot of the gunk before passing it through the filter. So certainly and clearly there's a lot of work going on here, as this is a inherent bottleneck.
Wonderful. Thank you very much. I really do appreciate all the questions that have been submitted, and like to thank very much our expert panel here. John, Rich, Conrad, thank you very much for your time and for lending your expertise and your knowledge for this session. Thank you very much for that. I'd like to also thank LabRoots and GIBCO, part of Thermo Fisher Scientific, for making today's webcast possible. Additional questions will be answered and published on Thermo Fisher Scientific's website, and you'll receive an email notification when answers are available. Before we go, I just want to let everybody know that today's webcast will be available for on-demand viewing through April of 2018. So you will receive an email from LabRoots letting you know that this webcast will be available for replay. Please share that announcement with your colleagues who may be interested that missed the live event today. Again, that's all. Thank you very much for joining us for this, and we hope to see you again at a later date. Thank you very much. Goodbye.